Hi class, let's talk about the discovery of DNA and how it's a double helix. So we now know that DNA is a double helix. It's got two strands that's looking like this, intertwined together, and the helical structure is from the bases and how they're oriented. Okay. So back in the early 1950s, there was a race to determine what the structure of DNA looked like, and the two people who were credited for this was Watson and Crick, these two people. They were awarded with the Nobel Prize to symbolize that they discovered the structure of DNA, but in fact, this is not 100% true. So they were the ones who collected and analyzed a lot of the data other researchers put together, but they did not actually perform any experiments. They just gathered information. Okay, so people who were actually contributing to this process, mainly it's this gal right here, Rosalind Franklin. Linus Pauling was also a major contributor, but this was very early on. So this is right before the 1950s. Okay, so Rosalind Franklin was an expert in X-ray crystallography. We'll talk about that experiment in our next slides. Working with Franklin was Maurice Wilkin. He took Franklin's work. So Franklin took some amazing images of DNA, and Wilkins found it on her desk and just stole it, took it right to Watson and Crick, and took it without any permission, and Franklin was not given any credit or recognition for her contribution. And then Franklin died in her early 30s, pretty soon after the discovery, and this was due to most likely from exposure to X-ray. So X-ray is radiation, and that's very dangerous for the human body. And she was not given any credit whatsoever. And she couldn't get recognition from the Nobel Prize because you have to be alive in order to receive a Nobel Prize. And she's dead. Okay, so let's take a look at X-ray crystallography. So this image represents X-ray crystallography. You might also hear it as X-ray diffraction experiment. So what happened is you have X-ray here. And this is a lead screen. So lead is actually, it prevents X-ray from passing through, most radiations from passing through. So there's a little hole. Some X-ray is able to penetrate and go through, and it hits the DNA sample. And once it hits that sample, almost like light shining through a window and hitting the glass. So once it hit the glass, it will refract. So it will diffract into rays. So there are multiple rays that comes out, and it will hit a photographic plate. So once it hit that plate, it will put an image, almost like taking a picture. So it will put that image using the X-rays. And the result looks something like this. Okay, so Rosalind Franklin, she did that exact experiment. And the photo that she got was this picture here. This is a very famous photo. It's called Photo 51. It actually telltales to the scientists that this is a double helix because the circle right here is the backbone. So this on the outside, the phosphate backbone that we see on the outside, that's what that black circle is. Okay. It also shows that DNA is very symmetrical. So it's like a circle. If you cut it in half, bisect it this way or this way, it's reflective. It's symmetrical. This tells you that the diameter of the double helix remains the same throughout. So it's not like it's narrow on the top and it gets really large on the bottom or anything. That means it stays the same throughout the entire structure. So the diameter stays the same. Okay. And with this, these black bars in the middle, those are actually the bases. And that's pretty cool. You can actually see the exact bases. So back when Watson and Crick were designing their model, they actually put these bases on the outside and they put the phosphate backbone on the inside. So that was totally wrong. Only with this image that Franklin did were Watson and Crick able to figure out that the backbone is on the outside and the bases are on the inside. Okay. This is a little something extra, but these bars are actually representative of distances. If you measure the distance between each bar, it actually tells you how far away the bases are. So there's actual measurement for this. So hopefully this explains a little of the history of DNA.